Hello, everyone. Welcome to another computer vision spotlight session here at ACE. My name is Mahdi Biparva. I'm R&D engineer at Sunny Brook Research Institute and also a computer vision stream owner here at ACE. This is my pleasure to host a great talk today titled Learning Permutation Invariant Representations Using Memory Networks. I'm excited to host Mr. Shivam Kalra in this amazing session. Shivam is a PhD candidate at the University of Waterloo. His supervisor is Professor Hamid Tizhush, and he's part of Chemia Lab. There are around 30 students in the lab focused on machine learning for digital histopathology. He's also a machine learning scientist at Huron Digital Pathology, and the area of his research is around image search, multi-instance learning, and active learning. Without further ado, let me welcome Shivam to our session. Hello, Shivam.
Yeah, hi Mahdi. Thank you for the introduction. And it's a pleasure to be talking about my work with ACE. So today, as Mahdi said, I'll be presenting this work that we have just published in ECCV, and it's called Learning Permutation Invariant Representations Using Memory Networks. The model that we have developed, it's known as Memory-based Exchangeable Model, or MEM. So today's talk, the outline is as follows. First, I'll give you a little bit of motivation why we wanted to develop this. Then I'll explain the problem definition, what we are trying to solve. Then I'll discuss the method, what we have developed, and then some of the experiments that we have conducted with our architecture. So traditionally, a machine learning model handles two types of data. The first one is fixed instance, fixed dimensional data, single instance, fixed dimensional data. So for example, an image, and you give it to a CNN, and it classifies that image into whatever category you specify. And generally, those images are of fixed dimensions, so 300 by 400 pixel, let's say. The second type of data is that could be handled through traditional machine learning is ordered instances of data, and generally of variable length. For example, you're doing sentiment analysis on a natural language sentence. And in that case, you can always use RNNs or some other machine learning technique that could take instances, but they're always ordered, or they have a temporal dimension into them. However, in many scenarios, especially in our lab also, our data was such that the labels were associated with input set, and they're unordered, and are also of the variable length. When you learn from such kind of data, when it's unordered and it's a set type of data, this is known as multi-instance learning, or MIL. So example is you have an input set, x1 to xn, and then you give it to MIL technique, which is a learning method, and you learn the label y associated with the entire set. So no matter how you permute the elements inside that set, if you still give it to MIL problem, it's going to be the label y. And the big part of MIL is that the permutation invariance should be encapsulated inside the model itself. It should not be augmented through the data. So for example, you have a set of handwritten digits categorized into two categories, even and odd. And I'm just showing different sizes. They could take different sizes. One could take 2 images, another in the same model could take 3 or 4 images, and so on. So many real world applications could be fabricated through this MIL scheme. And the reason we developed this method, MEM, is because our lab is mostly focused on representation of whole slide images. These representation could be either used for search, which is my field for PhD, or it could also be used for classification. If you have a good feature vector, it could be used for anything you want. So just to give you brief idea what is whole slide image and what is digital pathology. I have few slides for these field. So digital pathology incorporates the acquisition, management, interpretations of pathology information digitally instead of using traditional method. So traditional method would be looking at the microscope to analyze the slides. Instead of doing that, it's done digitally. So let's say you have a patient and you took a needle biopsy. So you create a glass slide with his or her tissue samples. So you have these glass slides, and then you give these glass slides, which are physical slides, to this whole slide image scanner, and what you get as an output is a digitized version of that glass slide. So it can contain the same resolution what you can visually see at the microscope. So this is a very large image, and this is known as WSI, or whole slide image. And this could be managed on a computerized platform, could be shared within the pathologist, and could be stored in a hard disk, and so on. So what is whole slide image? Like I said, whole slide image is a digitized version of a glass slide. And the data in WSI is very interesting. It's very different from natural images. It is arranged in a pyramid. So what you have is you have a base layer, which is the highest resolution that that microscope supports. So it is generally around 40x or 20x and so on. And they're very, very large. So it's around 50,000 or 40,000 pixel by 40,000 pixel, much larger than a natural image. Since this is a microscope, since this is trying to emulate a light microscopy, what you do is, what digital scanners do is they have multiple layers, so that when you're zooming in, when pathologist is zooming in digitally, they could, instead of going to the base resolution and then resizing, they could just directly go to the intermediate layer. So if my base layer is 40x and I want to see the structures of cells or tissue at let's say 10x, I could directly go to the 10x layer instead of resizing from the 40x 4 times and making it 10x. So they're structured in a pyramid. So it's a single thing. It's a single image file with multiple images in a pyramid structure. And usually, I should mention this, usually each file is around 1 to 4 gigabyte. So it's a very large image. So what are the challenges of whole slide image classification? First one is they're very huge images, so you cannot feed them directly to deep networks or, in fact, any traditional machine learning techniques also. And another problem is that in the pathology routines, the labels are generally generated for the entire WSI, entire whole slide image. And they are not annotated for a regional or a pixel level. So we do not have labels for patches inside that image. Instead, we have a label associated with the entire image. Also, like I said, there is a different magnification, and different magnification can encode different type of information. And resizing, if we say that we can just resize the entire image into something smaller that could be fed into CNN and then later out classified, this is not the best option. In fact, if you try to go take your technique and try to get the FDA approval, they will not do it, because this is something that is not considered good in histopathology. You should not be resizing the image. If pathologist is making a diagnosis at 20x, then your technique should also be able to take 20x and process that. So in this paper, our main motivation was that we wanted to formulate the classification of this large whole slide image as a multi-instance learning problem. So for example, you have this large image, which I resized to a thumbnail size. This is a very large image. And its label is papillary thyroid carcinoma. So one of the carcinomas or one of the cancer. So there is a tumor inside it. We don't know where it is because there is no pixel level or regional level annotations. So we just know that there is some carcinoma inside this large image. So what we did is we recently published these two papers, Yatixel and Pan Cancer Diagnostic Consensus. We published them in Medical Image Analysis and Nature Digital Medicine very recently. And we have developed a technique where we can systematically patch this large image into set of representative patches. So instead of dealing directly with this large image, you are dealing with this set of unordered patches. And by the way, each patch inside this, we call this mosaic, this set of patches, we call it mosaic in those papers. So this mosaic contains, let's say, roughly around 100 to 200 patches, and each patch is of size 1000 by 1000 pixel. So still you can generate, from one image, you can generate up to 100 to 200 images, each of size 1000 by 1000 pixel. Now what we want to do is we want to classify this mosaic, so set of patches, instead of this large image. So this, since we are dealing with a set of patches, the classification of WSI we formulated as a multi-instance learning problem. There are other important applications in real world for multi-instance learning. The another things that we tested for this paper is point cloud classification. We used one of the data sets that is quite popular. And the input for that would be the set of coordinates in a point cloud, and the output would be object class. Now, since it's the set of coordinates, so the order of the coordinates do not matter. Same thing, one more application is 3D shape recognition, where you can take the 2D images from different angle for a 3D object, and the output would be the object class. So again, the order should not matter. So you need to formulate the MIL such that when you are giving the input, the instances inside the input, the order should not affect the final output. If you can have that, you can utilize our model for learning from those type of input data. All right, so little bit about the problem definition. What is the problem that exists in the current machine learning models? So for MIL, the machine learning model requires two things, which are very important. The first is the permutation invariance. That is, the output remains same for any permutation pi of the element inside the input set. And the model should be able to process an arbitrary length input set. So some of the existing methods, for example, CNNs and RNNs, violate at least one of the above properties. So CNN violate both the properties. It cannot take arbitrary length. Does not have permutation invariance. Whereas RNN violates the first property of permutation invariance. It can still take the arbitrary length input. So just more mathematically, you are trying to learn a function f, which operates on a set x, producing output y. So example would be, let's say you're trying to find maximum of the set of handwritten digits. So you're trying to learn this f, that given 4 to 5, it do a regression and predicts 5. But at the same time, it should inherently be able to say that it's also 5 when you permute the elements. So 2, 4, 5 should also be 5. So this is a very important paper. It's called Deep Sets from a Zahir. It was published in 2017 in NeuroIPS. And this forms a basis of our paper that I'm presenting. And they said they have basically proven that if you arrange your neural networks according to the following equation, you can have a universal approximator of a set function. So remember the notation. So X is a set of input, and the X1, X2, and Xn are the elements inside that input set. So what you're doing is you're applying some kind of neural network phi on each element of the set. So they are weight shared neural network. So phi is one single neural network applied on in a iterative manner on each individual element. And then once you get those output of those neural network, you pool them. So you can do mean pooling, you can do sum pooling, you can do dot product pooling, or you can do other sort of pooling. Once you pool them, you pass that pooled features through another neural network or further nonlinear transformation. And if you can arrange your neural network according to this equation, you basically have a set approximator, any set approximator. So they prove it to be the universal approximator of set functions. However, what they did is they simply arranged according to this equation. So they simply arranged the neural network according to this equation, and they were not able to capture interactions within the elements. So we have extended, we have used the same equation, but we have extended it so that the attention, so that our a neural network could capture the attention within the instances, or it could calculate or compute the interactions that are occurring within the elements of the set. And in fact, we believe that many tasks, especially for multi-instance learning, can benefit from attention or these element-wise interactions. This is because not all elements of the input set contribute equally. This is specifically true for histopathology images, because there is a factor, that nuclear density, which could be a vital indicator for the prediction of a diagnosis. For example, if you look at the top 3 patches, they have higher nuclear density, which means that they have, according to pathologists, it might have higher probability of predicting the cancer than the bottom 3. So for a neural net, for a machine learning model, the first top 3 should be given higher attention, or should be attended more to predict the diagnosis than the lower bottom 3 images. So how did we incorporate attention? So I'll discuss our method, which is MEM, Memory-based Exchangeable Model. This is our method. It contains 3 components. So first is the feature extraction, where we pass the input set, and each individual element goes through this feature extractor, and we get another sequence of features, which is F, which goes through memory block. And then finally, we get a single condensed vector from the memory blocks, encoding the entire feature sequence. And then the single vector could then be passed to MLP for classification or regression. I'll discuss the memory block, which is the major component that we have proposed for this paper. It consists of memory units, and what it takes is an input sequence, and it gives a permutation invariant feature set as the output. So since it is also another set as the output, they could be sequentially connected to learn more complicated features, more complicated interactions, and so on. So memory block, like I said, consists of memory unit, which has a bunch of transformations. I'll discuss this, also the motivation why we are doing these transformations. And yes, going back to that equation. So since we want our architecture to be the universal approximator of set function, this has to also follow the same equation that was given in the deep set by Zahir in 2017. So like I said, the phi would be the feature extractor in this case. It is a neural network which is applied on individual element of the set function, input set. And then row and pool are basically memory block and MLP together. And in the paper, if you read our paper, we have shown why they are pool and row, why they follow this equation. So we have 2 proof that shows that our architecture can follow this equation. And then since it follows this equation, it is a universal approximator of a set function. So we have 2 key elements in that we have proposed in our paper. The first is the memory unit. It is a permutation equivariant layer that uses transformer-like self-attention to model element-wise interaction. And memory block uses memory units, and it is a permutation invariant sequence-to-sequence model. So it's important to know what is permutation equivariance and what is permutation invariance. So let's say you have an input set X or input sequence X, and you have an output also as a sequence, let's say Y. So something is permutation equivariant if you permute the elements inside the input X, and the same permutation is applied to the output sequence Y. So that is permutation equivariance. Whatever you permute inside the input element gets the same permutation applied on the output element. The permutation invariance is when you apply the permutation in the input set, but the output remains still the same. So that would be invariant to the permutation. So again, just to highlight that memory unit is a permutation equivariant and memory block is a permutation invariant. So memory unit, which is permutation equivariant model, we have three steps in this. So first is we embed the sequence, given input sequence, to memory vectors. And we are using the paper from Wetson Memory Networks, which was published in 2014. And the idea is that we want to transform the X into some embedding space. And this should be learned. So this is the parameter that is learned that embeds the X. So there is a parameter AI for each memory unit I. Once you embed them into memories, we use the self-attention, which is transformer-like self-attention, taken from the paper Attention is All You Need, published in NeuroIPS 2017. And this computes the self-attention over input. So if I give if my sequence is of n elements, I compute a attention vector, which is also of n elements, and each element signifies the probability value, which is between 0 to 1. So three steps are involved in the memory units. First, you embed the input to memory vectors, and then you use a transformer like self-attention, and then finally you get the self-attention over the input. Now we utilize this self-attention for our memory block. So memory block is permutation invariant. So why was memory unit permutation equivariant? Because see, this is a attention over an input. So if you permute the input element, the attention is going to permute in the same way because it's the attention over the same input, right? So this is permutation equivariant. Now let's jump to the permutation invariant part of our architecture, which is memory block, and this uses memory units. So there is a memory unit that I explained in the previous slide, and we have a bunch of these, M of these, inside a single memory block. And by the way, each memory unit learns its own embedding space. So they share the same input sequence, and then they embed them, whatever they learn, in individual memory unit. And then what we do is we aggregate these, we aggregate the final output through the bijective transformation. So bijective transformation is another neural network inside this memory block, which takes the X input and converts it into another sequence, C. This is also permutation equivariant sequence. And this is basically an autoencoder. So C is the deepest layer of the encoder. The idea is that we want to maintain the information inside X and the C, but C is slightly, what do you call it, compressed version of X. So once we have the C, we take the outputs from the memory unit, which are the self-attention, and we aggregate them. And what we get as the final output is the permutation invariant representation of the input sequence. So if your input is of n elements, of each element is of dimension f. F could be, by the way, multi-dimension also if it's an image. So it could be n cross h cross w, or it could be n cross f. So there are n elements of some dimension f. What you get as the output is m cross h. So m is number of memory units you have in that memory block, and h is the output that you are the zipped compressed version of the feature space. So if f is some image, it could be converted to h depending on your bijective transformation layer. And yes, so if you permute the element inside the X, which is n element, if you place them somewhere else, the m cross h, the x hat vector, stays the same because it is just the mean over the self-attention vectors, self-attention that you get from each of the memory unit. So this is our method, and we did few experiments. We did a lot of experiments actually. And so going back again, so here we have feature extraction and a memory block. And then you get, even from the memory block, you were getting a sequence out, if you remember. So there is a sequence, X hat is a sequence, M cross H. So you cannot give a sequence to a MLP, right? So you have to make it into a single condensed vector. So we we used 5 type of configuration to do that. So first one is that we can pool a sequence using the operators that were also there in the deep set paper. So we can do max, we can do mean pooling, we can do dot product pooling, or we can do sum pooling. And they have their own advantages. So max could learn some sort of identity. Some is more resilient to the if there is more elements inside the sequence, the sum is more resilient than the mean, because mean will just divide it by the length of the sequence. So it will nullify the effect of having more elements inside the input sequence. So everything has its own advantage. And we also used another interesting configuration for making it into single condensed vector. What we did is we we our last our last memory block can only contain a single memory unit, and we call this MB1. So if it contains a single memory unit, here the M would become 1, right? So it will become 1 over H. So 1 cross H. So it's a single condensed vector for the sequence N cross F. So this is another special type of configuration that we used. It's called MB1 in the paper. So we did experiments. We started off with the toy experiments, whether our architecture could even have a capacity to learn from for the simpler data sets. And then we applied to real world data set, and our main application was histopathological image classification. And we also applied it to point cloud. So I'll discuss some of the experiments that we conducted. So first is the sum of even digits. And this is a regression problem. Here the input is set of MNIST images. So you have, it's a handwritten images. So you have 4, 2, 5, and then the output is sum of only even digits. So 4 plus 2. So it has to ignore 5. The idea is that we want to exploit the attention mechanism of our model. And this gives an advantage to our model that it can just not attend 5, or it just can simply learn to not attend odd digits. And then it also only has to learn the joint probability of the even digits. So 4 plus 2 is 6, and it can do so. And we found that our model, especially since this requires attention, our model is performing better than the deep set. So these experiments are done such that the capacity of the model is similar, and we try to achieve the best results using both the models. And what we achieved is 94 for our model, and the deep set, I think maximum it achieved is 91, and we achieved the best one using max configuration. So next is the prime sum. And this is a classification problem. The input is set of MNIST images, and the output is true if sum of any 2 images is a prime. So here 2 plus 5 is 7 is a prime. So this again requires attention mechanism. It can exploit those attention or interactions that we have in our model, and can learn the joint probability in such a way. So here again, we achieved 94 using dot product configuration this time. And also we achieved the similar using MB1. In fact, MB1, we achieved almost similar every time. So MB1 is a very good configuration, and it can achieve similar to the best one in all the cases. Because sometimes the dot product, for example, in this case, the dot product is scoring 84, whereas in this case, it's scoring 94. And the best that we could achieve for the deep set is around 93 or 92. The next we did is sum of a set. So this may not require too much attention, because you're simply finding a single element inside the given set of images. So if you're given 5, 4, 2, you are just finding or regressing to 5. So this does not require too much attention. It does not require interactions within the elements, and it requires to only attend single image inside the set. So we achieved 95 here, and the deep set is also able to achieve more, in fact, more than us. And we found that it was able to do so because it is a simple identity mapping. So it could map each of the individual digits to some vector, and then it would learn the identity for that vector, which is a maximum. Then we also did experiment with counting unique images. We took images from the fashion MNIST in this case, and we have this set, and what the output should be is the unique classes that exist in this set. For example, I think there is 3 classes here. So one shirt, and then one pant and one boot. So yeah, this is what we did. And again, the results are very similar for DeepSet and our paper. It could again be that the attention may not be exploited well in this particular example. So now we have established that our method works. It can, if not bad, it can perform at least equivalent or better than the DeepSet, which is state of the art. And now we wanted to classify our histopathology images. So I already explained this. We used the approach that we published in the previous two paper in medical image analysis and Nature Digital Medicine. We used the same approach, and we patched a bunch of lung WSI, lung whole slide images, into a mosaic, which is again, mosaic is a set of patches. So in this case, there is this papillary thyroid carcinoma, and then we used our techniques to patch it into set of 70 patches, 70 patches. And now what we wanted to do is we used the lung whole slide images from one of the largest repository of whole slide images called the Cancer Genomic Atlas, TCGA. And we wanted to classify these lung WSIs into two types of lung cancer. So lung squamous cell carcinoma, LUSC, and lung adenocarcinoma, LUAD. So we in fact downloaded total of 2,580 whole slide images, out of which 1,249 belongs to lung adenocarcinoma and 1,331 belongs to lung squamous cell carcinoma. So just to compare with this seems like a very small data set, only 2,500 WSIs. But just to compare it with the ImageNet, this is actually equivalent to ImageNet. The total data that we downloaded is 2 terabytes. And we end up in extracting 2.5 million patches, each of size 1,000 by 1,000. But since we have the systematic technique of removing the patches, so we were able to make it less. So we were able to make it like, if you multiply with the 120-ish, we were able to make it around 100,000 or so. So this is what we did. We extracted the mosaic from each of the WSIs from lung. And then we give that mosaic, which is set of variable length patches, to our proposed model. And we were able to classify directly the entire mosaic. So we were not doing patch level classification or some other tricks. We were directly doing the classification of the entire set or the mosaic itself. And this is the accuracy we got. So we are here, and the MEM plus sum. So it's interesting to note that the sum achieved the best, not the mean. So sum is 84. We achieved 84% accuracy. And there are other state of the art, which are in fact published in Nature and very good journals, other state of the art, but they are using around 1,600 WSIs, and we are using 2,500 WSIs, so 1,000 more than them. And we are achieving very similar accuracy. And this state of the art is only using 338 WSIs. And one more thing that we found is that we performed five-fold cross validation on our data set to achieve 84.84, whereas other studies have used their own test and train split. So we cannot really justify whether the numbers are good or not, because we have performed full cross validation, whereas they have their own set of train and test. And we also found that the other papers have utilized some form of pathologist's opinion or annotations or experts' knowledge, whereas we have not done any of that. We have extracted the patches using an unsupervised method that we published, and then we simply used our MEM model to classify them into the lung cancer. And we were able to achieve equivalent to what they achieved. And just to compare with the DeepSet was not able to go beyond 77. Then we did the final experiment that we did is a 3D point cloud classification. We used a very popular data set for point clouds, and it's called ModelNet 40. It's published over here, and it contains 40 different objects embedded as 3D points, embedded in a 3D space as points. So we selected only 100 points in the same way that DeepSet did. for their paper, and we classified only 100 3D points using our model. So since they are 3D points in the Cartesian coordinate, so the orders should not affect anything. And this is the results that we get. So it's important to note that the instance size is what you are feeding to your network. So the other state of the arts who are feeding 100 points to the network, we scored uh, around zero point 0.1 improvement. So 1% improvement is there from other state of the arts feeding the same size. But the when you're feeding more, obviously you can uh, learn much better embeddings, so you can classify much better. So highest state of the art is 95, but it's using 32 raised to power 3 number of instances, whereas we are only using 100. So at 100, we are able to exploit a lot of instance interactions and stuff. So we are able to achieve this high. And the final conclusion is that we have proposed a permutation invariant network for set inputs. And we did it because there was an important application in the medical classification. And the results are active even for the other MIL tasks such as point cloud and simple toy datasets. So thank you.
Thank you, Shivam. That was a great presentation. I actually indeed uh, learned from your presentation. So we have a bunch of interesting questions that I would like to ask them from you. So one is that, so from what I understood, you mentioned at the beginning of your talk that the WSI are kind of image pyramids that are recorded from different resolutions. So
Yes.
did you use the patching across all the image pyramid, or you just took the highest resolution?
We took the highest resolution, and we wanted to take the highest resolution because this is the only resolution where everything is there. So if you want to, if there is some cancer which requires you to go individual cell mitosis and stuff, you can find those stuff in that resolution. And the type of structural, if you want to see the structure of the entire image, you can see it from the bunch of patches, right? So you still have a set of high resolution patches. See, structural level information could be extracted from the set in its entirety, right? So you have both type of information. You have a structural information through a set, and you have individual information through an individual patch.
Correct. But at the highest resolution, you most probably try to leverage the local uh, of information rather than the structure of the larger kind of larger size.
True. I would also like to add one more thing. So the two papers that I showed, we are in fact using lower resolution. So we are using 5x to extract the patches because they give you better information. You can process the image much faster. So we do use 5x to extract the patches, but we extract it at 40x or 20x.
Oh, okay, so that's kind of bridges to the next question I have. Can you a little bit elaborate on the policy that you implemented for extracting patches? So you very quickly mentioned that you have some sort of filtering to decide. So how do you decide to put aside some useless patches and just stay with the one that you are interested? What is the policy that you use?
Yeah, it's actually published in we call this a scheme or policy. We call it your Tixel. And the idea is that we do bunch of stuff. We do like a clustering. We do deal with the color spaces. We do bunch of ensembling of machine learning techniques to be able to come up with a systematic scheme that could extract these representative patches from the WSI. And if anybody is interested, you can just search my name and then a paper called Your Tixel. So Your Tixel is basically like a 1 gigapixel, right? Gigapixel and Your Tixel. So if you search that, I think it should be an open paper. And
Okay.
if you read through it, you can understand the entire technique that we have used.
Oh, great. So we have a number of questions on the live chat. So someone asked, what is the input to the, so can you explain what every single element in the set is? Is it pixels? Is it images? Is it a vector? How is it represented, the input
Yeah, that's a
elements?
that's a good question. And could I show my presentation? Do you see it right now?
I think it's gone now.
Yeah, let me show this and then just
Yeah,
yeah, you see it now, right?
right.
So the input is over here. You have oops. So this is the input, and it consists of n elements. So
Mhm,
it could be either image or it could be simply a feature vector. So f is not a single valued dimension. It could be n cross h cross w. So if it's an image, it could be n, which is number of images, and then its width and height and number of channels. So it could be
mhm,
anything. In our case, what we did is, since we are dealing with 1,000 by 1,000, and you cannot feed that many and train at the same time, so we extracted the features pre before feeding it to our memory blocks. So we used the dense net, which we also did in that paper, the Yortexel paper I mentioned. We extracted the features, and we are directly dealing with the feature of the images. So if you
great.
want to deal with a pixel value, you can do that too. It could be a pixel, it could be a feature, it could be an image, it could be anything. The last dimension does not affect anything. It's a single entity.
So it's actually the feature embedding at the top of the backbone, the feature extractor that you have. In this
Yes,
case, resonant.
yes, that's true. If I were to correct this diagram, if I were to make it more for our histopathology images, instead of feeding the input sequence, we feed the input feature sequence
Mhm,
of the patches.
mhm. So basically, the mosaic you mentioned, it's basically the mosaic is composed of single patches that you extracted and kind of filter out, and then you pack them, feed them into the network, and the network generates a feature embedding of every single element in the set, and then you
Yes.
feed it into your model.
Yes. So so you have a mosaic here, which is a set of patches. It's a real image, but instead of feeding the mosaic directly, what we feed is for each of the patch inside the mosaic, we extract the features,
Mhm,
and then we feed that feature set to our model.
mhm. Great. So, can you elaborate a little bit on the role of bijective transformation? So, how did you do any appellation study on um, the demand the space that the bijective transformer reduce the feature to? So, what is the role of it? How does it affect the performance and accuracy?
Yeah, that's a very good question. In fact, we did not do, unfortunately, any ablation study, but I can tell you what happened when we didn't use the bijective transformation. So what was happening is, sometimes the let's say you have an input sequence, right? And
Mhm,
sometimes the two elements inside the input sequence would map to a single point inside. If we were to not use a bijective transformation or an autoencoder, it would map to a single point, and then it would lose the information as the final output sequence.
mhm,
And it can do so if you're trying to find a maximum value, because it only needs to care about maximum, right? So it can collapse the information inside X to something, a single entity, which is still useful for the final classification that you're doing, but may not be useful for general purpose representation. So we used bijective transformation so that the information in X and the C is somewhat maintained. And this could be done through an autoencoder.
Mhm. Great. Did you use variable set sizes, or you fix them to some particular value?
Yeah, we did use variable set sizes, and we there is there is actually a very little trick how to do that. You can make anything variable in TensorFlow or any library that you use. So
Yeah.
you simply mask the gradient. So
Oh,
this is what
okay.
RMM does too. So they
I
have,
see.
in fact, now TensorFlow has a layer called masking
Yeah.
layer. So
Right.
you apply that layer before you feed it to the input, and then TensorFlow on backend, they automatically just ignore the gradient that is beyond that when the mask is set to 0.
Hmm.
So it's
We
simply
have
the description of gradient.
yeah, we have a similar thing in NLP too. So we do padding and packing, and we do unpadding and unpacking and unpadding. So
Right.
that sort of thing.
So on the
But
coding
so
side, you are feeding the fixed instances, but you are masking them, so it is a variable sized input.
okay, I
So
see.
we made sure that when we are calculating the mean, we don't do it like divided by dimension. Instead, we do it divided by the mask. How many ones are there in the mask, right?
Mhm. And another question. How many memory blocks? So you used only two memory blocks?
Actually, that is the hyperparameter that we did experimented with. So
Hmm.
for the toy experiments, we were able to do it with like 4 or 5, 3 or 2, and so on. But for
Hmm.
the bigger data sets, we had to not just use multiple memory units inside memory blocks, but we also had to use multiple lots of memory blocks to
Hmm.
be able to capture the good features.
Okay. So was it that on large data sets, as soon as you increase the number of memory blocks, the performance kind of improved?
It was not magically improved. What we found is that we had to play a lot with number of memory blocks, at the same time number of memory units inside each memory block.
I see. And somebody asked about the computation, the cost, the computation cost of the memory, the model that you proposed. How much overhead did you add to the network?
Yes, it is computationally expensive because you are doing a self attention, which is a matrix multiplication to itself, to its
Hmm.
transpose.
Hmm.
So the computation is definitely involved, and you're doing this for each memory unit. So if you have m memory units, you are doing it m times.
Hmm.
There is a computational involved, but fortunately, there are also libraries that gives you more optimized routine for calculating the self attention these days.
Hmm.
So you can do like a matrix to matrix transpose multiplication in a batched way. So we
I see.
did the computational, we did some efficiencies, but only at the coding level. And there is, at the mathematical level, there is complexity involved.
Hmm. I think, let's
But I
see.
would add one thing that the histopathology data set that we experimented with, it's almost like an ImageNet. So we fortunately had two V100s, each of 32 GB, so we were able to do it. But there is a computational definitely involved in the memory units and the memory blocks.
Did you use batch size, large batch sizes? How did you manage to do the um, batch
Since now you are dealing
training?
with a set, right? So there is already 100 images inside each set.
Yeah,
So we end up in only using a batch size of around 6 to 8.
I see.
So each thing inside your input would be another 100 images. So
Yeah,
it is already like 600 images inside
right.
if you're feeding as a
Yeah,
single image. So
that's huge. Um, do you have any intuition on the application of your approach in NLP?
in fact, memory blocks. Sorry, the this memory vector paper, the first paper called Memory Networks, was done only for question answering. I think it was question answering for NLP. So the idea of memory is that you can keep the sentence memory. There is a memory for every word. So if there is an Eiffel Tower or there is an Paris, they have a same sort of memory. That was the idea.
Mhm.
And the first application of the memory networks was for NLP. So I've not done myself experiments with the NLP, but I believe since it's the ideas originated from the NLP, it should have applications in NLP. And this is something that if anybody is willing to contribute to, that would be a great work, I guess.
Yeah, I believe so, especially in at the level of document. So once we have a document of various paragraphs and sentences and that sort of so huge amount of language um, text data, then we
True.
can definitely apply this sort of approaches for
I'm sure attention
classification.
could benefit from now. You don't just have memories, but you also have attention on top. So
Mhm. Yeah. So that's great. I really enjoyed the talk. One of the topics of my PhD research was on attention, and whenever I see application of it on vision applications, it's always fascinating, and I always enjoy. And so I would like to appreciate you for the time that you dedicated to the presentation. And I also want to thank the audience for joining us. I hope everyone enjoyed the talk. So
yeah,
thank
thanks a
you
lot, man.
and have a good day.
You
Take
as
care.
well.